And so in today's lesson, we're going to be talking about dimensionality reduction with an emphasis on principal component analysis. And so there are a number of learning objectives of this activity. And so first, in terms of data science, we want to understand the principles of dimensionality reduction, including the two main features of feature selection and feature extraction. I want you to understand the utility of dimensionality reduction and how it can be used to visualize complex data sets and transform data for downstream applications. Primarily machine learning is what we'll be doing in this course. In terms of Python learning objectives, I want you to understand how to filter, subset, and transform pandas data frames. And we'll also be using scikit-learn to actually run the principal components analysis and to do some downstream calculations. And then we'll be using seaborns to plot our principal components of our data sets. And then in terms of plant biology, the goal is to understand the types of data frames that come with gene expression data, and then to visualize and interpret a complex gene expression data set using a principal components analysis. So first off, what is dimensionality reduction? And so dimensionality reduction is the transformation of data from a high to a low dimensional space while still capturing the intrinsic dimensions or the meaningful properties of the original data. And so in biology now, we're collecting larger and larger data sets. So this could be genetics or genomics. So in case of gene expression, you might have tens of thousands or even hundreds of thousands of gene expression values and lots of different tissues. So you have these massive expression matrices. Maybe it's phenotypic, where we have different shapes of leaves. Maybe you have field data where you're collecting tons and tons of images and point clouds. Maybe it's physiologic data or developmental data. But increasingly we have these massive data frames, and it's very difficult to work with these and use them in downstream applications. And so we want to find a way where we can take, tens or hundreds or thousands of columns and rows and condense it down to something more meaningful without losing the intrinsic dimensions of the data set. And so in general, raw data can be very space intensive, and it can contain a lot of low information data points. And so we want to kind of filter out the low quality data so that it doesn't inflate our downstream analyses. And in general, processing high dimensional data is very computationally intensive. And so if you're using machine learning, you wouldn't want to input features that have, 20,000 genes as input features, because it would make any calculations that you did prohibitively time intensive, especially considering that most of them are not going to be that informative. So this is a way to go from very large complex data down to meaningful smaller data sets that we can then process in downstream applications. And so let's consider a gene expression data set as an example. And so a typical gene expression experiment in plants may have between 200,000 or between 20,000 and maybe 100,000 or more expressed genes across dozens or even hundreds of tissues, time points, and samples. So we have this very large expression matrix, and so if we envision a matrix like this that has 20,000 genes, where each row in the data set is going to be a different gene, and so these would be essentially all the genes in the genome. And then we could have any number of samples, so here we're just going from S1 to 100, so maybe we have 100 samples where we're looking at different tissues, different developmental time points, maybe stress versus non-stress, some sort of circadian experiment, whatever you want. And then here each number corresponds to a gene expression value, and if this were RNA-seq, this could be RPKM, reads per kilobase per million reads, or some sort of similar metric, where basically the larger the number, the more that gene is expressed. And so if we have this expression matrix here, so let's say it's 20,000 by 100 are the dimensions, this is really difficult. So you can't just look at this and draw any meaningful conclusions. We can't look at the replicates and say how similar are they to each other. We can't look at different tissue types or different time points and say how does this data look. And we couldn't really put this in any sort of downstream application, such as machine learning, where maybe we had a training set, we wanted to predict from gene expression alone what our tissue is or what a stress is. So we want to use some sort of technique to reduce this massive complex data frame into something more meaningful. And so this is a ton of data, lots and lots of data points. And in general, a lot of this data is not going to be meaningful. So you'll have a lot of genes that have no or low detectable expression. You'll have a lot of genes that are highly correlated with each other. So you might have a couple thousand genes that are expressed at the same time, at the same level. We would say those things are co-expressed. So maybe having all those individual data points is not that meaningful. And so the main question is, can we reduce the complexity of this data while still capturing the variation between samples? And also, is there a way that we can draw meaningful conclusions from a reduced complexity data set? So if instead of having this massive data frame, we have something a lot smaller, can we then look at that and get some sense of our data before we do downstream analyses? So those are kind of the main questions we have with running these types of analyses. And so in terms of dimensionality reduction, there are sort of two steps. So the first one is feature selection, and then after that you would do feature extraction. And so we're not going to go into too many details about this. You can do this in other courses, or you can research these independently. But in terms of feature selection, so this is a way that we would take our raw data set and transform it in some way to either remove low quality data, to parse different subsets of the data, or figure out some way of removing low quality or low information data. And so there are three sort of ways that we can do this. We can use wrappers, we can do data filtering, or we can do embedded. And so wrapper methods use a predictive model to score feature subsets, and so they would take a subset of the features or a subset of your data matrix and use that to see how it predicts other parts of either predicts some sort of phenotype or predicts some property of the data, and then you might filter the data that way. This is very computationally intensive, using a lot of predictive modeling, and there are lots of approaches that you can do here, but this is very intensive and is not applicable to every data set. We can also just do some filtering, and so filter methods use a proxy measure instead of the error rate to score a feature subset. And so a good example of this is you could apply some sort of Pearson correlation across the matrix and see how different elements of the data set are correlated with each other. And you can remove the data based on some metrics. And generally, this is less computationally intensive, but it's less accurate than wrappers. In terms of data filtering, we can also do some simpler filtering. So we can do some preprocessing where we do some sort of feature scaling. So maybe we do some sort of z-score or log transformation, or you can do some sort of mean normalization. And again, this is not the best way, but it's very low intensity, and it can be a good way of kind of filtering out some of the data before you do feature extraction. And then the third way is embedded. And so embedded methods perform well. They perform feature selection as part of the model construction. And so an example would this be like using Lasso to build a linear model. This can be really great. It's less computationally intensive as wrappers, but in general it also tends to be a little bit less accurate than wrappers. So these are kind of this is sort of the first step where we want to select the features that we want to do the dimensional reduction and kind of filter our raw data frame. And then the second component is actual feature extraction. And so this is where we're actually going to be reducing it into a smaller number of dimensions. And so what we'll be talking about today, and this is sort of the most common linear-based approach, is a principal component analysis. So we'll talk about that here. And then other linear approaches for feature extraction include linear discriminant analysis and multidimensional scaling. We won't talk about those today, but they kind of similar to principal component analysis. And then we also have a couple nonlinear feature extraction approaches, such as TC and UMAP, or the T-distributed stochastic neighbor embedded, or uniform manifold approximation and projection. So we're not going to get into these. These are a little bit more complex statistically, but you can use these different programs or these different algorithms or mathematical models to try to reduce the complexity of your data frame. So Today we'll be talking about PCA, but you may encounter a couple other of these in the course. And so we won't really get into the math or the linear algebra behind PCA. If you want to learn more about this, there are tons of resources out there that you can go there. But instead, we'll just be talking about how to run this, but kind of give you sort of a general visual representation of how a principal component analysis works. And so PCA is sort of the main linear technique of dimensionality reduction that you'll encounter. So it's the simplest, it's probably the least computationally intensive, and it's used in many different fields. And so you can use it for visualizing data and also as like an input for running machine learning algorithms. And so PCA works by reduction of high vector space through lower orders using projection. And so it can be used to reduce a large, n-dimensional array of complex data into something less complicated. And so here's sort of an example here. So let's try to reduce this three-dimensional plane into a two-dimensional plane. And so here we have this set of data that's plotted along three axes here, the x, y, and z axis. And so the question is, how do we go from this three-dimensional down to something a little bit simpler? So we'll put it into a two-dimensional space. And so first we have a plane. You find a plane that captures most or all of the information or variability between the samples. And so here we kind of find this plane here that captures along two orthogonal axes here that captures most of the data. And then this data is then projected onto a new set of axes with reduced dimensions. And these would represent the principal axes or principal components. And so here we've taken these three dimensions. We have created this plane here, and then we projected it onto two dimensions. So this is sort of a representation of how PCA works. And so you can apply this to many higher dimensional data sets and reduce their complexity as well. And so there are tons of uses of a principal component analysis and other dimensionality reductions. These are some that are kind of going to be relevant to our course, but you can use it for all sorts of data. So the first is to reduce the complexity of a large data frame into meaningful simplified values or principal components that you can use as input features for machine learning. And so again, like with expression data, instead of having 20,000 features that you would input for machine learning where each feature would be an individual gene, you could run a principal component analysis on the whole data frame, and then you could input maybe a couple or maybe a dozen or a couple dozen principal components as features for machine learning. And so if you're incorporating a lot of features, this will give less weight to expression-based ones, and it's also a lot less computationally intensive. And we can also use it to approximate the relationship between different samples across multi-dimensional space. And so maybe you wanted to look at replicates of data. So again, if you're collecting perhaps phenotypic data, physiologic, biochemical, gene expression, some metric of the genome, you could then run a principal component analysis, plot the data in a couple of the principal components, and then see how your different replicates correlate with each other. So in general, if they're replicates of the same data set, you expect them to be closely related to each other in a principal component analysis. So we can also look at interactions or correlations between variables and different data types. So this can be really great if you're collecting multiple different types of data and you want to see how those variables might be correlated with each other. We can also use it for gene expression, and so you can use it again to transform it from machine learning. We can use it to get a sort of visual representation of the expression space in which things tend to be like each other. You can use it again to look at replicates. So if you have 3 or 5 replicates of, let's say, leaf gene expression, you can see if they're correlated with each other or if they're not, and that can give you a general sense of the quality of the data that you've collected or that you're working with. And we can also use it to look at genetic relationships between individuals or species. And so we can feed it a matrix of variance, so differences between different accessions or different individuals, and then we can plot a principal component, or we can calculate the principal components and then plot them for those. And so we'll actually be doing this in one of the later notebooks. So this is just kind of a subset. There are tons of of really interesting downstream things you can do with principal component analysis. And so here's an example of a PCA that was run on gene expression data. There's kind of a lot going on here. We have a couple actual dimensional reduction techniques that we've used here. And so in terms of data, what we have are 3,000 gene expression data sets. So this was data that was analyzed in this Plants and Python course in 2020. And so we collected about 3,000 gene expression data sets from the short-read archive from about 60 different species across angiosperms. We mapped them against common orthologs from all these species, and then we looked at the expression values for this matrix that was essentially 5,000 orthogroups by about 3,000 individual samples. And each one of these data points is a sample here. And so we're showing here the first two principal components, and then each of these points is colored by the plant family here. So brown is one plant family, red is another plant family, purple is another plant family, etc. And what you can see is that these first two principal components are largely separating our enormous data set by plant family, where principal component component 1 is separating sort of these brown ones across PC1. PC2 is separating this red family, so this is the Poaceae or the grass family, separating it from some of the other groups, and then together these separate these really nicely where we can see these fall into separate groups. And I'm just showing a couple other dimensionality reduction approaches that were applied to this data set. So this is MDS, or multidimensional scaling, and you can see, similar to the PCA, it's separating across these two MDS1 and MDS2, separating them out pretty nicely by family. And then we also have TSNE, which this is a nonlinear dimensionality reduction based approach, and you can see this is separating it very cleanly by plant family. And so this is sort of a starting point. We could do this to look at individual samples, but in general this is kind of a nice way to run this analysis, visualize your data, and then see how your data is kind of looking in a very large sense. And so we could then do downstream machine learning on this. We could do some other transformations, but it's a nice way to get an initial visualization. And so we'll be using the module scikit-learn to run a principal component analysis on a set of gene expression data. And so we're just going to scratch the surface of what scikit-learn can do, but it's a really powerful package for various statistical analyses, machine learning, dimensionality reduction, predictive modeling, and other types of data analysis applications. And so scikit-learn can do tons of things. We're just going to do a couple things in this course, but definitely check this module out. It's really powerful. And so what we're going to do is we're going to import a data frame in pandas of gene expression. We'll transform it, and then we'll run a principal component analysis on it, and then we'll plot it using Seaborn. And so the first thing we need to do is we need to import our modules here. So we'll import NumPy, and we'll import pandas, and this will be for manipulating our data frame. And so then we need to actually import our expression data frame, and so I will provide this for you. And so we'll create our variable here of expression data frame equals, and then we'll use pandas to read it in, read_CSV. So this is going to be a comma separated format, so we'll use pandas.read_CSV. And then we will just provide it the actual name of the file, and so make sure that when you import this data that you are using the correct file path. Here this is in the same directory as my notebook, but just make sure they're in the same directory. And then we will also, for this data frame, we will index the columns, and this is for some of the downstream things that we will do. So we'll simply set the index using the first columns here, and then we will create our data frame, and then we can visualize our data frame. So let's do X, let's call our data frame, and then we'll do .head and see what we have here. And so what we have is each, the first column is going to be our gene names here, and so this is data from the important Ethiopian cereal teff. So this is expression data for 10 different tissue types. So seeds at different developmental stages, leaves, roots, flowers, etc. and we ran this through the gene expression pipeline that we talked about in the previous lecture in this notebook. And the first column is going to be the genes, so there should be around 23,000 genes. And then the second each of the other columns here will be an individual data set. So this is going to be this first stage of seeds, replicate 1, replicate 2, replicate 3. So we have 3 replicates for each tissue that we have here, and then we have 10 individual tissues. And so this is the data frame that we're actually going to be manipulating this activity. And so we can also look at the shape of this data frame. Shape. And what this will tell us is that it is 23,000 rows long, so there's 23,000 genes, and then there are 31 columns, and the first column is gene name, so there are 30 samples. And so this is a really massive data set, so we're going to run a PCA to reduce the dimensionality of this, and then to look how the individual replicates correlate with each other and how the individual samples correlate with each other. And so the first thing we need to do here is to filter our data frame. And so with gene expression, you have a lot of genes that basically have no or very little expression. This is the sort of sparse, low quality data that we want to remove, so this is part of the filtering step. So we're basically going to remove all of the data that has less than RPKM of 10, or reads per kilobase per million base pairs, if you remember. So this is just sort of an arbitrary number where the higher the number is, the more that gene is expressed. And so what we'll do is we will first we'll just print the shape of our matrix before filtering, and so this will tell us basically the genes before filtering, and then we'll also remake our data frame, and then that will tell us the genes after filtering. And so we will first create a list of genes from that satisfy this criteria, where the sum of the expression for the genes in each row is greater than 10. So this would have all the genes that have an RPKM of greater than 10. And then we will create a new data frame from this, where we will use this .loc. And so this will basically return all the genes that we've created in, that satisfy this first criteria. And then we will create a new data frame here. And then if we run this, basically what happens is we start with 23,000 genes before filtering. We basically maxed out the low expressed genes by only creating a variable with the genes that have higher than 10, and then just rewriting a new data frame with only those genes. And then right now we have a data frame that went from 23,000 down to 18,000 genes. And so these are genes that have basically some level of expression. And so by removing the ones with low expression, we're removing low quality data points that probably wouldn't be very helpful in our analyses. And so also our gene expression data, if you were to look through more rows, you would see that the values will range from an RPKM of less than 1 to an RPKM of greater than 10 or 20,000. And so we're looking at really 5 or 6 orders of magnitude variation between these numbers. And so if we were to use these numbers for doing dimensionality reduction, because there's so much magnitude difference, the genes with really high expression would have a lot more weight in determining the principal components compared to the genes with low expression. And so we want to log transform these, or transform them using z-score or some other metric, so that the numbers are more comparable to each other. So the low genes will still have lower expression than the high genes, but instead of it being a value of 1 versus a value of 10,000, it would be a value of like 1 point something versus 10 point something. And so we're going to transform these by log2, each number in our data set, and that will have it more comparable so that we get a little bit better analysis here. And instead of doing a log transformation, you could also transform by z-score, which is where you're sort of using the mean normalized data, and then going from values of 0 from 1 based on that. But just for simplicity, we're going to do log2. And so we're going to use these two sets of Boolean logics here with a nested for loop. And so basically, we're going to create our first for loop of 4 column in, and then basically it's going to be for each column in our expression data frame that we created. and then we're going to have this if statement that if numpy dot is subtype, so this is going to detect if it's a float or an integer. So if our data type is a number, basically, do the following thing. And so it will look through each column of our data frame, and it will basically look for each number, and then if that comes back as true, then it will do the following thing. And so in our data frame right now, we might have some zeros, and if you do a log transformation of 0, you get undefined. And so we want to take all those zeros and we want to add +1 to everything in the data set. So this won't really change the data a lot, but it will basically remove all of our data that has zeros, and then we can actually log transform it. And so what this first statement will do is it will iterate over this entire data set, and it will look for every number. And when it finds a number, it will do this action where it will basically add 1 to that number. So then we go from, this will take all the zeros, make them 1, it will add 1 to everything, and then we can actually log transform it. And then we have basically the same Boolean statement here, where we have the same sort of nested loop system, where here instead we're going to look at for every number in every column, if the data type there is a number, do the following, and instead of adding 1, we're actually going to log transform it. And so we will use this numpy.log, and then call our data frame, and then call basically each value. And so this will iterate over the entire data frame, and then for each value it will basically log transform it. And so if we run this, the first time it's going to add 1, and then it's going to log transform it. And so if we actually look at our data frame here, you'll notice that the values are a lot more in line. So instead of having numbers that are like ranging from less than 1 to maybe a couple hundred or a couple thousand, 10,000, we have all of our numbers are going to be between around 1, less than 1, and the highest number we might see is 10 or 11. And so you can see here a lot of these numbers are between 0.5 and 2 or 3, but if you look across the whole data frame, you'll see a lot less variation. And so this transformed matrix will be a lot better for actually running a principal component analysis, because the numbers will be more comparable to each other. Okay, and so the next thing that we want to do is we're going to transpose our data. And so right now, the way we've set up our data frame is we have each column is going to be a separate sample, and then each row is going to be a separate gene. And so if we ran PCA the way it runs right now, we would be basically running a principal component analysis using every gene, but we want to reverse that. So we're going to transpose the data where we'll switch the columns and the rows, where now each row will be a different sample, and each column will be a separate gene in that sample. And so we'll simply use this function .transpose, and we will transform our data set. And then so now we've kind of inversed it. So each row in our data set is going to be a separate sample, and each column is going to be a different gene. And so now, instead of it being 30 by 1800, it's now 1800 by 30. And so for each one of these, there's 1,800 different columns. And so, 18,000 different columns. And so now we can actually run a principal components analysis on this. And there are a lot of functions that NumPy can do, but it usually takes a considerable amount of time and some kind of finagling to go from your raw expression matrix or your raw whatever data frame you're importing into one that you actually want to run your statistics on. Okay, so now that we have our data frame that's in the correct format, we can run the PCA on it. And so we will import our module here from scikit-learn.decomposition. We'll import the PCA module. And so first we'll set our variable here where we're going to set the number of components. And so when you run a principal component analysis, if you were to just make a simple plot, you might want to plot the first two principal components. But those first two principal components are only going to capture some percentage of the variability in your data set. So if you're using this for machine learning, you might want to set this to the first 10 principal components, maybe the first 20. In our case, since we're just going to plot the first two, we will just set this as 2. And it will only output the first two principal components. And and then you can set this at whatever number you want. So this will set basically the number of components to calculate, and so we will create a variable here of real underscore PCs, and then we will use the function PCA .fit underscore transform, and then we will feed it, PCA is going to tell it the number of components to run, and then we will feed it our expression data frame, and then we'll basically from that create another pandas data frame where the data will be this real PCs variable, and then we will name the first two columns as principal component 1 and principal component 2. So if you were going to instead run this with 3 principal components, you would then have a third data column here that you would call PC3, or for however many principal components you were going to run here. And so this should run relatively quickly, just take a couple seconds, and the amount of time depends on the complexity of your data matrix and the number of principal components that you're going to output. And so what it's done right here is we've given it our data frame, and it's going to be using some linear algebra to calculate the first 2 principal components in our data frame. So let's look at what we have here. And so the first question you might have is how much variation is explained by the first 2 principal components. And so we can use this function here where we say PCA dot explained underscore variance underscore ratio, and this will tell us basically the percentage of variance that is explained by the first 2 principal components. And so if we run this, it basically will output the first principal component, or PC1, is explaining 36.7% of the variation in our data set, and then the second principal component is explaining 13.5. And so you might have a question like, is this good? I would say it's pretty good. In general, if you think about a gene expression matrix, you're looking at biologically dozens or hundreds of different processes and pathways, maybe thousands that are occurring in a plant cell. You're looking at lots of different tissue types, and then we are heavily transforming our expression matrix. We're getting rid of genes without a lot of data. We're not capturing the full cascade of genes in this organism. So I think this is actually pretty good. So our first principal component represents about 37% of the variation, and the second is about 14. So that's pretty good. If you were to run more than 2 principal components, let's say 3 or 5 or 10, this would output however many components that you ran. And so the first 2 PCs, they'll always go decreasing the amount, and so really the number of PCs that are meaningful depends on your data set. And so we'll actually visualize this using Seaborn, but what you could do is take these numbers and put them in as input features for machine learning or linear regression or whatever downstream analyses you were interested in. Okay, so the first thing we need to do before we plot this is we need to actually add back our sample names, and so right now we just have a data matrix in pandas that has 2 columns where it has the values of principal 1 and principal 2, and so what we want to do is basically read this CSV that I've supplied to you. This is called tef underscore names, and so this is basically a list of all of the different names we give each sample. So you could make this the same as before, or you could create it whatever names you want. You can also probably craftily pull these from the previous data frame, but we won't get into that today. And then we will take this new data frame that we've created, and we will concatenate it with our old data frame using, so we'll call our variable here the final real data frame, and then we will use this function pandas.concat. So this will basically merge 2 data frames. And so when we do this, both of our data frames need to be of the same dimension, so they need to have the same number of rows, or else we'll get an error. And so we'll basically merge these 2 together of the sample names. That's going to be first because our first column should be the names of the samples, and the second will be our principal component data frame. And then we can run head on this as well and see what our data frame looks like. And so now we had the first 2 principal components, which were output in this real underscore PCs underscore data frame, and then we have our name here. And so what this is these are 2 values in this vectors of PC1 and PC2, and then for each one of these samples here. And so now we have this nice data frame. Let's plot it. And so we will plot these first two principal components using Seaborn. And so we first need to import Seaborn module here. And then here I'm just going to very simply plot this graph. You know, you can get really fancy with Seaborn. You could color them by replicate. You could color them by tissue. Here I'm just going to give them a random hue for each of the individual samples. And so we will do sns.lm plot. We will plot PC1 and PC2. These are going to be our first two PCs. We will give it a height of 10. The data that we're going to feed it, this data frame that we've created, the hue will be colored by tissue, where each one of our samples will be a different color. And then we'll set this flag as false. And then if we run this, sometimes you get an error when you run Seaborn, I don't know why, so let's try to run it again. And then we have this plot here. And so if we look at our plot, again, I haven't colored this super well. You could change the colors and have it a little bit more meaningful. But we basically have PC1 on the x-axis, PC2 on the y-axis, and then we have each of our tissues here. And if we look at the distribution of our data, we can see that these three samples here are highly clustered, and so these are correlating to this D root replicates 1, 2, and 3. And so these are going to be roots under desiccation from, or under extreme drought from the sample. We have 3 replicates here. These are going to be 60% relative water content, or kind of a mild drought. And then we have other data distributed here. And so this is sort of just test data. Ideally, what you would expect to see is that each of the 3 replicates would be highly clustered with each other. But you can see here that the roots tend to be over here, and then leaf tissue tends to be down here. And so we kind of just get a sense of what the data looks like and what it's showing here. And so again, you can use this on any data type and run a principal component analysis and then visualize it and see how much variation the different principal components are explaining. Okay, so just to summarize here, what we've learned so far today is, in terms of data science, understanding the principles of dimensionality reduction, including feature selection and feature extraction, understand why we would want to use dimensionality reduction for our data, and how it can be used to visualize complex data and transform data for downstream machine learning and other applications. We learned how to filter, subset, and transform data frames using pandas in Python. This is really important. Your data frame will almost never be in the format you need it to be, so doing transformations is really helpful. And then we use scikit-learn for principal component analysis. And then in terms of plant biology, we visualized the gene expression data set, we transformed it, and we can gain some understanding based on how the data looks in a spatiotemporal way. And so for the remainder of this lesson, what you'll be doing is running RNA-seq analysis on your own set of data, and then you will be running a PCA on your data just to see how it looks, and that would be used as a starting point for any downstream analyses that you would be doing.